The mitotic spindle, often called the spindle apparatus, is really one of the most important parts of mitosis because the spindle apparatus is what will be responsible for moving the chromosomes around during mitosis. The spindle apparatus is part of the cytoskeleton. It's made up of microtubules, which are polymers made up of proteins, which are called tubulin. So that's where the name microtubule comes from. And this is a very dynamic structure. So it can be very quickly built and taken apart. It is assembled at the centrosome. So the centrosome is the structure that these will actually grow out of. So the centrosome is building them. And as those centrosomes start to move apart from each other during early prophase and make their way to opposite sides of the cell, the spindles are going to be actually growing out of them. So we say that the spindles begin to form during prophase. So they would begin to form during early prophase, and that would really continue throughout later prophase, or what's often called prometaphase. And the big job of the mitotic spindle is to control chromosome movement during mitosis. So if the mitotic spindle does not work properly, the risk is that the chromosomes are not going to be separated as they should be, and ultimately we won't end up with an even number of chromosomes on the two sides of the cell. So it forms during prophase, it's going to line up the chromosomes during metaphase, and then ultimately it's going to separate the chromatids during anaphase. And when it separates those chromatids, it's going to be specifically separating the sister chromatids. Keep in mind that the sister chromatids are identical to each other. And so when we actually pull these sister chromatids apart from each other, we have the exact same thing moving to two poles of the cell. Now we want to keep in mind that this structure where the spindles actually attach to the chromatids is called a kinetochore, and you would have one kinetochore per chromatid. Now, another thing that we haven't talked about much is what's called cytokinesis. Cytokinesis, by definition, is division of the cytoplasm, but basically we're talking about the rest of the cell. So where mitosis was division of the nucleus, this is division of everything else. And if we are to actually end up with two completely separate cells, we will have to divide the rest of the cell. Now this is going to start during late mitosis, basically during anaphase. And usually by the time we finish telophase, cytokinesis is going to be done completely. So it isn't a process that takes a real extensive amount of time. The cytokinesis process will be different depending on whether we're talking about an animal cell or a plant cell. And this is because the outer covering of those cells is definitely different from each other. As far as mitosis is concerned, there is no difference between a plant and an animal cell. The nucleus has the same structure to it, and all of those phases that we've already discussed, they would be exactly the same. So we'll talk separately about the animal cytokinesis and the plant cytokinesis. So if we first talk about the animal cytokinesis, keep in mind that humans would be considered animal cells, so this also applies to them. And we call this process in animal cells cleavage. Now the outer covering for an animal cell is that extracellular matrix, which is very flexible. So in this case, during anaphase, those cells are going to start to elongate, so it won't be a spherical cell anymore. And then we're going to have some proteins called myosin. And then there will also be some parts of the cytoskeleton called microfilaments. And these are going to make kind of a ring or a rubber band around the middle of the cell. These will slowly contract, and as they contract or they shrink, we're essentially going to pinch this big cell into two separate individual daughter cells. That's the cleavage process. Now we can see this actually happening in this little animation here, or it's really not an animation, it's a movie of this process. So these are actually sea urchins cells, and we're going to see them go through a couple cleavage events. Now you would have nuclei inside of those cells, but we're just not able to see them because they're not really stained for that particular purpose. So as we watch them go through this second cleavage here, notice that those cells are just simply pinching apart from each other. So this is all because we have that contractile ring that actually forms down the middle of the cells. It shrinks, and as it shrinks, it's actually going to pinch the cells in half. Now if we look at this process in plant cells. In plant cells, you have a cell wall surrounding them. And this cell wall is a very rigid structure. So because it's a rigid structure, we're not going to be able to just simply pinch the cells in half. That won't work. So the approach is going to be a little bit different here. Rather than pinching the cells in half, we're going to just construct a new cell wall down the middle. So this is going to take place through the formation of a structure called a cell plate. Now, it's important to remember that a cell wall, or a cell plate for that matter, is made up of a molecule called cellulose. And cellulose is a polysaccharide, but it's a structural polysaccharide. So in order for this to happen, we're going to have little vesicles that form down the center of the cell. So here we can see those little vesicles kind of forming down the middle. Keep in mind that vesicles are basically little membrane bubbles, and in this case these are going to contain cell wall material inside of them. So if I just draw a couple of them here, this is essentially what we have forming down the middle of that cell. These individual vesicles are going to grow, and as they grow, they will merge into each other. So we'll end up with something more like this as they continue to grow and they merge into each other. So we're still going to have cell wall material or cellulose inside this larger vesicle at this point. That's what we call the cell plate. So the cell plate is this large piece of cellulose which is down the center of the cell. Eventually it's going to span the entire cell, and it's going to run from one cell wall down to the other cell wall. Now the significance of having this cell wall material enclosed in a vesicle to start with is that not only have we built cell wall material down the center of these two cells, but we've also lined that cell wall with plasma membrane on either side. Because not only did we need cell wall down the middle, we also needed continuous plasma membrane around each of these new daughter cells that we have. Now as far as the size of the daughter cells is concerned, they will be approximately half the size of a parent cell, but they'll pretty quickly grow to be that normal cell size. The overall goal of mitosis and cytokinesis is to produce two genetically identical cells. And genetically identical cells are basically clones. So that is what we're talking about here. We're talking about cloning cells. We're producing cells that are exact copies of each other. And the type of cells that we're producing is going to be somatic cells. So in this mitosis, we're going to have a parent cell that is a somatic cell, and it will produce two daughter cells, which are also somatic cells. And somatic cells will be identical genetically. Now, if we contrast this a little bit with prokaryotic cells are most definitely not going to be able to go through mitosis and cytokinesis. That really should not be a surprise, keeping in mind that mitosis is specifically division of the nucleus. And a prokaryotic cell does not have a nucleus that it can actually divide. So these are going to have their own way of reproducing, and this is going to be called binary fission. Binary fission is going to be a much simpler process than mitosis and cytokinesis, and it's also going to be a much faster process. So to start off, if a prokaryotic cell is going to replicate, then it, just like a eukaryotic organism, needs to copy all the genetic information. So this means that the chromosome has to be copied. Now keep in mind that when we're talking about a bacterial chromosome, it is a circular chromosome. Okay, so they have basically one big circle of DNA. It's a double helix, but it's connected around into a circle. And this is going to be just sitting kind of in the inside of the cell in a region that we call the nucleoid, but that nucleoid is not really separated from the rest of the cell. So we're going to copy that big circle of DNA so that we end up with two big circles of DNA. Then the cell is basically going to attach one of those circles on one side, and it's going to take the others and attach it to the other side of the cell. At this point, as the cell begins to elongate, it's going to basically pull apart those two circles of DNA, so that now you're going to have one side of the cell that has one copy, the other side of the cell is going to have an exact copy as well. Then, very similar to the cleavage process, we're just going to have those two cells pinched apart to make two new cells. Now, this is going to be called asexual reproduction in prokaryotic cells because we are making exact copies. It is a much faster process because we don't have a nucleus. We don't have to worry about taking apart the nucleus. We don't have to worry about rebuilding the nucleus. And because we're only dealing with one chromosome, we don't have to build a spindle apparatus and carefully attach the spindles, pull the chromosomes apart, and then ultimately take those apart as well. As far as time goes, this whole process is going to take about 20 minutes. So from start to finish. So that's the copying of the DNA as well as actually splitting into two new daughter cells here. If you compare that 20 minutes to what we have with a eukaryotic cell cycle, for a eukaryotic cell, it's about 24 hours or one entire day. So obviously a much faster process, and this is why bacterial reproduction is much faster than eukaryotic reproduction. So you want to keep the major differences of eukaryotic cell division in mind with regards to prokaryotic cell division. If we now talk about the regulation of the cell cycle, the cell cycle is something that definitely has to be regulated. In other words, we don't want cells to continue to spin around this cycle and make new cells if we don't actually require new cells. So if we don't need new cells, there's really probably not going to be any room for those new cells. We also don't want the cells to progress from one stage into the next if they're not truly ready to do that. Because if that happened, then we would end up with cells which really aren't going to be exact copies of each other at the end of all of this. So throughout the cell cycle, we have points which are called checkpoints. And these checkpoints are basically times when the cell is going to stop and kind of take account of what's going on and determine whether the cell is really ready to go into the next stage or whether we really even need to be going into the next stage at all. There will be molecules present in the cytoplasm whose sole job is to regulate these checkpoints. So they will be making the decisions as to whether or not the cell gets to go into the next phase or whether it gets to spin around the cell cycle rather than sit in that G0 stage. So we have two different types of regulatory molecules or classes of regulatory molecules that are involved in this process. The first ones are called cyclin-dependent kinases. Now we'll learn, starting here, and you'll see it very commonly in the next few chapters, that kinases are a type of enzyme. And enzyme names we'll see do end in ACE or ASE. And we'll see kinases quite a bit, and kinase enzymes are going to be enzymes that basically phosphorylate other molecules. So in other words, they're going to be adding or removing phosphate groups from molecules. Well, these cyclin-dependent kinases, when they do their phosphorylation, it will be activating or inactivating some other proteins that we have present inside the cell. And these cyclin-dependent kinases, which we'll call CDKs for short, they're usually going to be present at constant concentrations. So at all time points during the cell cycle, there will be CDKs around. Now, it wouldn't make a lot of sense for them to constantly be activating certain molecules and inactivating others because there wouldn't be any regulation in that. So this is where the second group of molecules comes in. And these are called the cyclins. The cyclins, they control the activity of the CDKs. So they're going to actually bind to the CDKs, and when they bind or attach to the CDKs, this is going to flip the on switch for the CDKs. The cyclin concentration fluctuates all the time. So this means that even though the CDKs are always present, they will not always be turned on because there won't always be a cyclin attached to them. If we look at this picture down here at the bottom, MPF is an example of a CDK. And we're showing a line representing the MPF activity because if we did draw a line for the MPF presence, it would just be a solid line right across the top of there because the CDKs are present in constant concentrations. If you look at the line for the cyclin concentration, notice that it goes up and then it drops off, and then it goes up again and then it drops off. So there are times when we're not going to have any cyclin present inside the cell at all. Notice that when the cyclin is present, then we also have some activity for our MPF. But when there is no cyclin present, the MPF activity really drops off. If we look at this in another way, here we're looking at a particular checkpoint during the cell cycle. We're looking at end of G2, beginning of mitosis. So we would be deciding to go into mitosis. So we have this CDK, which is going to be all by itself, and it will be inactive. We also have a cyclin that just got produced. So if those two are both present inside the cell, they are going to bind together, and when they bind together, this will turn that CDK on, which if we turn it on, that means that it's then going to be able to activate some proteins, and then it will activate some other proteins. So it's going to do this through the attaching of phosphate groups to these molecules. Now if you look at where we're at during the cell cycle, we're getting ready to go into mitosis. So the type of things that it would be activating would perhaps be things responsible for condensing the DNA, because keep in mind that we need to condense the DNA as soon as we get into mitosis. We could also have things activated that need to basically build the spindles. So we're going to be activating proteins that we will need very shortly when we get into the next step. And we want to inactivate things that we're basically done with, that we don't need anymore. And if you think about G2, G2 was really all about double checking. So we can get rid of some of those double checking molecules or double checking enzymes and inactivate them. If we do get through this checkpoint, then the cell is going to take apart the cyclin. And when that cyclin is actually degraded, then our CDK will still be present, but it's not going to be working anymore. Now, a lot of times students question, why do we even need that second molecule, the cyclin, to really turn the other one on and off? And one big reason for that is that the CDKs are a lot larger than the cyclins are going to be. So perhaps not as obvious as it could be in this picture. But since the cyclins are a smaller molecule, it's going to be less time and energy intensive to kind of produce those, take them apart, produce them again, take them apart. Because the CDK is such a large molecule, it would take a bit of time and it would take a bit of energy to be constantly remaking that particular one. So this is going to be a much quicker, simpler on-off switch than remaking the CDK every time we need it. Now keep in mind that these are two big classes of molecules that we have inside the cell. We do have a lot of different CDKs and we do have a lot of different cyclins, and they are going to operate at different times during the cell cycle, and they will also have different target proteins that they're activating and inactivating. Okay, so the CDKs function by phosphorylating substrate proteins that will affect particular steps in the cell cycle, and they're really going to be responsible for us moving from one stage into the next. Now this is one way that cells are going to basically regulate themselves. So the cyclins and the CDKs are involved in self-regulation. We also need mechanisms for cells to regulate each other, so to regulate neighboring cells, for example. And this is where growth factors come into play. Growth factors are going to be proteins that are released or secreted from certain cells, and then these can be sensed by the neighboring cells, and that sensing will either cause those cells to divide, or it may actually inhibit their division, so it'll tell them not to divide. Examples of this are going to include hormones. So if we just look at one example where growth factors would be used, this would be in what we call density-dependent inhibition. And density-dependent inhibition is basically saying that when cells get crowded, they're going to stop dividing. So if they're crowded, they don't have room or they don't have the resources to support additional cells being produced. So there would be no sense in producing them. If you look here in this top picture, we have a Petri plate, which has a number of cells kind of scattered out on it, and there's lots of space there. So the cells are going to divide, or what we would call proliferate. So they're going through the cell cycle until basically they fill up all of the space available. Once they're all touching each other, they are secreting hormones, which they're going to sense, and they're going to know that they need to stop dividing. At that point, they would be sitting in G0. Okay, so when they get to this point, they're not actively dividing, they're sitting in G0, just waiting to see if they're going to get the signal that now we need new cells produced. So if you were to go in and actually scoop out or remove some of these cells, those neighboring cells would see or sense that there is some additional space there, and that would be when they're going to then jump right back into the cell cycle. So they would enter right into G1 of interphase, and they would go through the cell cycle to produce the cells they need to actually occupy that newly available space. So here's just another picture of it. Normal cells, if given the room, they're going to proliferate, or in other words, go around the cell cycle until they fill up all the available space. And then when they don't have any more space left, they're going to become quiescent or quiet, which means they're going to be sitting in G0. So at that point, we're not going to get any new cells produced. So basically, if we get an injury, cells that were in G0 are jumping back into the cell cycle to replace those damaged or injured cells for us. So cells do have a number of mechanisms in place to make sure that they're not producing cells if they don't need them, and also to make sure that if they produce cells, it is because they need them and they're going to get exact copies. If we talk about what happens when cells do not regulate their cell cycle properly, this is really where cancer cells come into play. Cancer cells are cells that do not respond to the normal control mechanisms that are in place. And those control mechanisms that would be in place would be things like the CDKs, the cyclins, hormones, okay, those ways of regulating themselves or neighboring cells regulating them. So they're not responding to the density-dependent inhibition that we mentioned or other types of inhibition. And what this means is that you're going to get cells that are just kind of out of control and they continue to produce new cells. So we say that cancer cells have uncontrollable cell division. And if we have uncontrollable cell division, then basically we end up with tumors or masses of cells that we did not want before. If you look in this diagram, we can see that a normal cell is going to have parent cells that divide to produce daughter cells. That will happen all the time. This is something that really is happening pretty much all over our bodies. And there are times when cells will become damaged due to a mechanical injury, so something actually hitting it, or this could be a chemical injury, but in any case, they're getting damaged in some way. If a cell gets damaged and we're not able to actually fix that cell, then that cell is supposed to undergo a process called apoptosis. And what apoptosis is this is defined as a programmed cell death. So basically a self-suicide. Okay, so we have a suicide of the cells. They're programmed to kill themselves if they are so damaged that it's just so abnormal and they can't carry on their normal functions. This is ideal for the cell because if that cell dies off, then our normal cells can divide and produce a normal cell to take that cell's place. We would not want a damaged cell to replicate because with the process of mitosis and cytokinesis, we end up with the exact same thing at the end. So a damaged cell would only lead to the production of more damaged cells. So with cancer, this is exactly what happens. You have this damaged cell that does not die off as it should, and instead it produces more damaged cells. and then those are going to have the same error that the original cell had, so they're going to produce more damaged cells. And before you know it, you have a mass of abnormal cells that are dividing uncontrollably. That would be cancer. Now, there are different ways to really classify tumors. We can have what are called malignant tumors, and we can also have what we call benign tumors. In this picture, we have an example of a tumor in some breast tissue. And any tumor is going to start as basically one abnormal cell that did not die off. Okay, so if you have an abnormal cell and it goes through the division process, it is going to produce additional abnormal cells, and we're going to then have a tumor produced. That tumor can grow because these cells are going to continue to divide. So in the first two pictures here, we have this tumor that is growing, and at that point, that tumor would be what we call a benign tumor. That's because all of those cells are right there in that initial location. Those are fairly easy to control because if we had surgery take place and we went in there and we removed that tumor, if we were sure that we got all of those abnormal cells out, then there would be no lasting problem there. However, if this tumor grows more, some of those cells tend to break off. And if they separate from the mass of cells, they can enter lymph vessels and blood vessels, and they can basically move to other locations in the body. When they do move to other locations, they basically stay there, and we get what's called a metastatic tumor. This is because that one cell that happens to land somewhere else in the body is going to continue to divide, and now we have a tumor in another location. This is what's called by metastasizing. We say that these tumors, they can be called malignant tumors because they are going to impair the functions of one or more organs. It's much more difficult to control this type of a tumor because we're never quite sure where all it has spread to, because it's going to take a period of time before it's really going to become apparent in that new location. It has to reach a certain size. We want to be familiar with what are cancer cells, the problems within those cells, which is essentially that those cells are not capable of controlling their cell division, so they're not responding to the normal control mechanisms in place, such as hormones or growth factors, CDKs, and cyclins. And then we do want to understand that there's different types of tumors, such as benign and malignant.